Welcome, dear students. After having learnt about the conductive apparatus in the physiology of hearing, now let us move on to the next step, that is transduction of mechanical sound energy into electrical impulses. And this is mainly done in the organ of Corti. So first let us study about the organ of Corti. This organ of Corti is situated on the basilar membrane. It is present all through the length of the basilar membrane, and we can study this organ of Corti by looking at this picture. This is a magnified image of the cross section at basilar membrane. So let me label this picture for you. This what we are seeing here is the scala vestibuli. And this is scala tympani. As we all know, the scala media is triangular in cross section. And separating the scala vestibuli and scala media is the Reissner's membrane. This is the Reissner's membrane. Separating the scala media and scala tympani is the basilar membrane. And on the basilar membrane is the organ of Corti. This is the organ of Corti. And this entire structure that you are seeing here is the organ of Corti. It is seated on the basilar membrane. Now let us see what is there on the organ of Corti. What constitutes the organ of Corti? First is the tunnel of Corti. What is the tunnel of Corti? This is the tunnel of Corti. The tunnel of Corti is lined by inner and outer rod cells, and it is filled with a fluid called as cortilymph. So this what you are seeing here is the tunnel of Corti, filled with cortilymph. And next we have hair cells. Hair cells are the most important part of organ of Corti because they are the actual receptors of sound, and they are the ones which convert sound energy into electrical energy. So we have two types of hair cells. Actually, these are the cells with stereocilia present on top of them. So under low magnification, it looks like hair, and hence we call them hair cells. And there are two types of hair cells, inner hair cells and outer hair cells. If you can see in this picture, these are the outer hair cells, the ones I am drawing in orange. This is an inner hair cell. So first let us study about the features of inner and outer hair cells, and then we can come back to the rest of the organ of Corti. So these are the differences between inner and outer hair cells. If you look at the cell numbers, inner hair cells are around 3500 in number, whereas the outer hair cells are much more, and they are 12,000. The inner hair cells are arranged in a single row, whereas the outer hair cells are present in 3 to 4 rows. The shape of inner hair cells is flask shaped, whereas the outer hair cells are cylindrical in shape. And the nerve supply of inner hair cells is mainly afferent, although a few efferent fibers are also present, whereas that of outer hair cells is mainly efferent and a few afferent cells are also present. And inner hair cells develop early, whereas outer hair cells develop late. And the function of inner hair cells is to transmit auditory stimuli, whereas that of outer hair cells is to modulate the inner hair cells. They do not have a direct role, they modulate the function of inner hair cells. And in response to ototoxicity or noise-induced damage, the inner hair cells are highly resistant, whereas the outer hair cells are sensitive and they are easily damaged. So they are the vulnerable type of cells. Outer hair cells generate otoacoustic emissions, whereas inner hair cells do not do that. So these are the differences between outer and inner hair cells. Going back into the picture, you can see multiple cells here, and these are of course our outer hair cells. They are arranged in 3 to 4 rows and they are cylindrical in shape, whereas the single inner hair cell that is marked here is flask shaped and it is present in a single row. This is broadly the structure of organ of Corti, and we also have a few supporting cells. All these cells that are seen here, these are the supporting cells. And names of a few supporting cells are these supporting cells are present between the outer hair cells and inner hair cells, and some of them are Dieter's cells, Hensen cells, cells of Hensen, cells of Claudius, and Boucher's cells. These are a few supporting cells. And finally, the last structure is the tectorial membrane. This is the tectorial membrane. What is the tectorial membrane? It is present on top of the hair cells and it is a gelatinous matrix. And it has delicate fibers in it. It is made up of delicate fibers and it overlies the hair cells on the organ of Corti. This is important because the shearing force between the tectorial membrane and the hair cells will stimulate the hair cells and eventually cause impulses and action potential. So this is the structure of organ of Corti. What we are seeing here is one single cross section, and this is present all through the length of the basilar membrane. It extends from the base of the cochlea to the apex of the cochlea. After having understood the organ of Corti, let us look into exactly how transduction of sound occurs. We have heard about part 1 of story, where the sound has been picked up and collected by the auricle and then it travelled into the external auditory canal, struck to the tympanic membrane, and via the 3 ossicles it has come to the footplate of stapes. And now let us see what happens from the footplate of stapes, from the oval window. So we stopped at the footplate of stapes or the oval window. What is present immediately after the oval window? It is the scala vestibuli. The scala vestibuli contains fluid inside it called as the perilymph. Now these vibrations, the sound vibrations, are travelling in the perilymph. And in the perilymph is in contact with the bone above and with the Reissner's membrane below. The bone is of course a hard structure, but the Reissner's membrane is delicate enough, it can get affected by the fluid, by the vibrations in the fluid. So, the sound vibrations reach the Reissner's membrane. Plus, they are being transmitted all through the scala vestibuli, and at the helicotrema, the same fluid becomes the scala tympani, and so these vibrations travel that way also, and finally the basilar membrane is depressed. Basilar membrane is depressed. So the sound waves in the scala vestibuli are transmitted via the Reisner's membrane. They come into the scala media, and then the basilar membrane is depressed, and then the basilar membrane is depressed into the scala tympani, and then what happens is the secondary tympanic membrane at the round window bulges out. The secondary tympanic membrane at round window bulges out. This is what is happening. But what is important to us is this step. Movement of the basilar membrane. Because when the basilar membrane moves, the organ of Corti situated on the basilar membrane also moves. And this all functions as a single unit. And so, there is a movement seen on the hair cells also. And the tectorial membrane is a rigid structure. It doesn't move along with these. So, the shearing force between the hair cells and the tectorial membrane is produced. And this distortion of hair cells will stimulate the hair cells. Let me write this down. When the basilar membrane moves, there is a shearing force between the tectorial membrane and the hair cells. So, the movement of stapes is transmitted to the cochlear fluids, and this will move the basilar membrane and the tectorial membrane differentially and sets up a shearing force between them that will bend the stereocilia between the tectorial membranes and hair cells and bends the stereocilia. And this bending of stereocilia is very important to us. Why? Because when the stereocilia are distorted, let us see what happens. Distortion of stereocilia will lead to potassium influx. And potassium influx will cause depolarization of the cell, depolarization of the hair cell. And this depolarization will lead to calcium influx. And calcium influx will cause release of neurotransmitter. So this is exactly where the mechanical energy, mechanical sound energy until the shearing force between the hair cells and tectorial membrane, until distortion of stereocilia, it was mechanical energy, and now because of potassium influx, it has been converted into chemical energy, and this chemical energy, because of the release of a neurotransmitter, will result in initiation of action potential in the auditory nerve fibers. So finally, it is converted into electrical energy. Initiation of action potential in the auditory nerve fibers. The hair cells are connected to nerve fibers, if we remember. And so, release of neurotransmitter will result in the initiation of action potential in the auditory nerve fibers. So, an important point here is, generally in any cell, it is the sodium influx which causes depolarization, and it is the potassium efflux which causes repolarization. But a very special feature in the hair cells is that potassium influx is causing depolarization. Generally it is sodium influx, but here it is potassium influx, and this makes it a potential question to be asked in the examination. Then now we have come to the end of part 2 of our story. After being conducted till the stapes footplate, now we understood how it undergoes this entire process, and the mechanical sound wave is converted into an electrical impulse. But there are a few more things that we must understand. One is, which sound wave affects which part on the organ of Corti? What do I mean by that? A sound wave, depending on its frequency, reaches maximum amplitude on a particular place on the basilar membrane. Now, the basilar membrane is present right from the base of the cochlea to the apex of the cochlea. And we have learned this while learning the anatomy of inner ear also. The length of the basilar membrane increases as we go towards the apex. And higher frequencies are heard at the base and lower frequencies are heard at the apex, and this is very, very important. So let me write that down. A sound wave depending on its frequency reaches maximum amplitude on a particular place. On the basilar membrane. And it stimulates that segment. This is how the brain differentiates high frequencies and low frequencies. So every frequency has one particular location. A frequency of 1000 Hz reaches one particular place. A frequency of 10,000 Hz reaches another particular place. Yes, and it stimulates that segment of the basilar membrane. So what we know is, higher frequencies usually reach their maximum amplitude at the basal turn itself, whereas the lower frequencies reach their maximum amplitude at the apex. This entire theory is called as traveling wave theory of von Bekesi. It is the name of the scientist, von Bekesi. So what does the traveling wave theory of von Bekesi say? In the fluid of cochlea, in the cochlear fluid, these sound waves are traveling like ripples in a pond of water, and the lower frequencies attain their maximum amplitude at the apex of the cochlea, at the apex of the basilar membrane, whereas higher frequencies reach in the basal turn itself. This is the traveling wave theory of von Bekesi. It explains the frequencies of sounds above 2000 Hz. Above 2000 Hz, this is what happens. But to explain about the differentiation of sounds below 2000 Hz frequency, we have another theory called as frequency principle or Wally principle. What is this frequency principle or Wally principle? It explains about the sounds which are below 2000 Hz. According to this theory, the electrical impulses equal to the sound frequency. Electrical impulses equal to the sound frequency. What does this statement mean? It means that if at all, for example, it's 1000 frequency sound, 1000 nerve impulses are created, and that's how that sound is transmitted. This theory is applicable to low frequencies up to 2000 Hz. It is explained by the Wally principle. More than 2000 Hz, it doesn't work that way. If suppose there is a 5000 Hz sound, 5000 nerve impulses are not sent to the brain. Those are explained based on the traveling wave theory of von Becquerel. Now, these two principles together explain pitch discrimination, that is how the mind is able to differentiate between different frequencies, and both of them together are called as duplex theory of pitch discrimination. They are called as duplex theory of pitch discrimination. Duplex theory of pitch discrimination includes traveling wave theory of von Becquerel and frequency or Wally principle, which explains less than 2000 Hz, and traveling wave theory explains more than 2000 Hz. So, A plus B together is the duplex theory of pitch discrimination. So, after this, we need to learn about the electric potentials inside the cochlea. We have various electric potentials in the cochlea. What are they? The first one is the endocochlear potential. Endocochlear potential. It is present in the endolymph, which resembles the intracellular fluid present in the endolymph is present in the scala media in the membranous labyrinth. So, this endocochlear potential is a direct current. It is a direct current that is present in scala media. How is this generated? It is generated by the stria vascularis. Remember, the scala media is triangular in cross section. It has Reissner's membrane on one side, basilar membrane on the other side, and on the third side there is stria vascularis that is responsible for the secretion of endolymph also. And this stria vascularis, by the action of sodium potassium ATPase pump, creates this endocochlear potential. Now, this endocochlear potential is a source of energy. This is present during resting times also, not just when the hair cells are stimulated by sound. Normally also, endocochlear potential is always present. It's like a battery that stores energy. Yes, this is about endocochlear potential. And next we have Cochlear microphonics. Cochlear microphonics. Cochlear microphonics are alternating current. Endocochlear was direct. This is alternating current. So, in response to the sound stimulus, the electrical resistance at the tip of the hair cells changes, and this allows flow of potassium through the hair cells. It causes potassium influx, as we have learnt before. And this potassium influx will cause voltage fluctuations inside the cell, and those voltage fluctuations are called as cochlear microphonics. So, what is cochlear microphonic? In response to sound stimulus, what happens? The electric resistance. Normally, there is some electric resistance at the tip of the hair cells. This electric resistance changes because of the sound stimulus. And when the electric resistance changes, it allows potassium influx into the cells through the hair cells. We have learned that before also. Distortion of stereocilia will cause potassium influx, and that will cause depolarization of the cell. Same thing here. The potassium influx will cause voltage fluctuations inside the cell. Voltage fluctuations. These will give rise to cochlear microphonics. Endocochlear potential is present during rest also, whereas cochlear microphonics occur only during a sound stimulation. Next, we have the third type called as summating potential. What is summating potential? It is also a direct current like endocochlear potential, and it is produced by the hair cells. This summating potential can be positive or negative. And summating potential is important in the diagnosis of Meniere's disease. Finally, we have the fourth type of electric potential called as compound action potential. The above three were produced in the cochlea, whereas the compound action potential is by the auditory nerve fibers. It is from the auditory nerve fibers, and it follows all or none response. Either there is a stimulus and an impulse is generated, or there is no impulse at all. There is no partial response, all or none. The compound action potential produced by the auditory nerve fibers follows all or none law. So these are the four potentials which are known as electric potentials in the cochlea. With this, we complete the second part of our story, that is transduction of mechanical energy into electrical energy. In mechanism of hearing, there were three parts. The first step is conduction of the sound from the external environment to the inner ear, and the second step is transduction of mechanical energy into electrical impulses, and the third step that we are going to learn in the next video is conduction of electrical impulses to the brain, and that is via the auditory pathway. So in the second step, we have learnt about the organ of Corti. It's important to understand the structure of organ of Corti and also the differences between inner hair cells and outer hair cells. All in all, the inner hair cells are present in a single row. They're flask shaped and they're less in number, only 3,500. They're resistant to ototoxicity and to noise-induced trauma, noise-induced damage, and they develop earlier and their nerve supply is mainly afferent. If you remember one side of the table, the other side of the table can be extrapolated. And then we also learnt about two theories of pitch discrimination. They are the travelling wave theory and frequency or Wally principle. Together they are called as duplex theory of pitch discrimination. And finally about electric potentials in cochlea. Only cochlear microphonics are alternating current. Endocochlear and summating potential are both direct currents. Okay, let us learn about the auditory pathway in the next video. Thank you very much.